Welcome to another BioSolveIT webinar, or it's actually not correct. It's not a webinar, it's more of a live design session. So welcome everybody to the first iteration, the first version, the first episode of What's the Drug? And yeah, the question is, what is what the drug? And to be very transparent, even we don't know. That's at least what I would say, because it can be had anyways. Before we dive into this whole procedure, maybe let me introduce you, at least the people who are new to this platform, to the stream guidelines. You are muted by default to avoid any audio feedback on our side. This session will be recorded and also uploaded to our YouTube channel, which is youtube.com/BioSolveIT tutorials. And this time you are highly encouraged to interact with us using the chat box. So if there's anything you would like to comment, address, or maybe even like send a smile string to us, use the chat box and we will try our best to make it as dynamic as possible. I'm a little back ahead. So what's the drug? So the flow of this stream, actually, we don't have a script. We never know what's going to happen, but we have kind of like a set in mind, or at least some milestones we would like to achieve. We are always starting with a PDB. This PDB can be anything, any structure, any target class. It can be GPCR, it can be kinase, it can be whatsoever. I personally hope for a GPCR structure. Sometimes, or maybe in the future, we'll also work with APO structures, depending on whom we will invite and who would like to participate here. And we are facing a challenge. So basically, anybody from our team, from BioSolveIT, will be sitting here talking to random people from the internet or from the science community. And we will be presenting a challenge. Basically, given a PDB, people will ask us design a ligand with picomolar affinity, at least according to our scoring algorithm. Maybe also replace a scaffold, find something to escape patent, I don't know. Or even things like maybe suggest three novel lead structures that are also commercially available. Anything can happen. It depends on the use case and depends on the mood of the people or the guest of the session. We will be having a time limit and try to achieve as much as possible within this particular time frame. And again, this is a community thing. So basically, this is where you can kind of like dive into our mindset. This is where we can dive into your mindset. And for us diving into your mindset, we need actually you sharing your minds and your thoughts with us. So please use the chat box, interact with us, comment. And if you don't want to comment, then please at least enjoy the show. And for today, I would like to introduce the guinea pigs for this format. It will be me. My name is Alex, and I'm from BioSolveIT. I'm working as a marketing director here. My background is I'm a pharmacist, and I worked on GPCRs during my PhD program. And now I'm here, and I'm willing to be the one facing the challenge today. And now I hand over to the challenger, somebody from within our own four walls, as I would call it, a person who maybe will be very nice to me, hopefully. Marcus, maybe you introduce yourself as well.
Thanks very much, Alex. Yeah, so pleased to virtually meet you all out there. This is a leap into cold water. So by design of this series, we did not study books on the stuff that we are going to do. But instead, we would like to create a situation in which you and we are from time to time, when a project starts and you get acquainted with the structure, you find flaws, pros, cons, weaknesses, opportunities, and stuff like this. So my background is also chemistry, theoretical chemistry, and I did a minor in bioinformatics. And I would like to challenge Alex today with a structure that is a co-crystal structure that we would like to embark from. And since this is our first challenge, again, the focus shall be on getting acquainted with the structure and then modify it in such a way that it makes sense, perhaps grow into a sub-pocket, etc. We will see on the fly. So also me, I don't have a script in mind, which is the script itself, to not have a script in mind. Alex, let's get acquainted with a structure that is 8QQG. So what I picked is, I picked the structure that caused a little bit of a of a communication wave recently, so it's a relatively new structure, a BRAF structure with an inhibitor that is not yet FDA approved. To the best of my knowledge, the structure dates back only a few weeks. And the first challenge would be that you and we jointly, of course, I mean, this is not a school setup scenario, that we first look at the structure, find out where are interactions, is the structure valid at all, what do we know about it? So what we're talking today, Alex?
Yes. So I assume you already mentioned the PDB, but maybe repeat it.
That is 8QQG.
8QQG. So what do we have here? We have a BRAF, so it's a kinase, no GPCR today, and it's in complex with exarafenib. Okay, let's dive into it.
So for those of you that are not familiar, this is our CISA suite of tools, or the CISA dashboard, if you wish, which is meant to be a very interactive tool for those that are not necessarily specialized in computer chemistry, computational chemistry. So Alex, back to you.
Okay, so I assume this is the carpet we are working with today. And by pressing on the spacebar, I will zoom in. And so this is exactly what I expected. This is a kinase, at least it looks like one to me. And we have several crystal structures, several chains. So basically what I always tend to do here is always compare. So it seems like this is already a pretty decent compound. So we, at least according to our calculations, it has a very high binding affinity. So we cannot even display it in our normal settings. And this is also reflected by the fact that every atom here contains a grain sphere. So basically every atom favorably contributes to the overall binding affinity. Maybe just go through and go through the structure here. So basically from what I know of kinases, this looks like the hinge binding motif here with the gatekeeper residues in this area. What's surprising to me is actually the structure that is supposed to bind in the hinge binding area, which is a morpholine. This is comparatively uncommon, I would say. You usually have like aromatic structures for those areas. I'm quite surprised, to be fair. So this is a very uncommon motif for hinge binders.
Alex, if you could zoom out a little bit, it seems like this might be a trimer. So perhaps we verify and we can check if there are any differences, if the structures are all the same. They all are WJ9. So this seems to be a chain B, and this seems to be a chain C compartment.
Yeah. So if we can also zoom in on them, it's basically just an ensemble from the crystallization process.
First thing also to those that are not so familiar, this is only a computer, yeah? And that means that we can overshoot, undershoot in our binding affinity estimation. Here we overshoot a little bit, but perhaps we also understand, given the visualization, we may understand why there are so strong contributions to the affinity. But we'll see as we discuss. Yeah?
Mm-hmm. So what else is here? We also have the chlorines, since we are now talking about what is co-crystallized here. The structures are all the same. No differences in the protonation states as well. What I noticed on the first side, though, is namely this little fella here. So this is for this area. So this is actually a solvent-exposed area where a lot of things are allowed in kinase design. Here we see some flexibility. So especially we have this cute little hydroxy group. And in this area, we see an intramolecular H bond that forms, which is not present, at least for the B chain, as far as I can recall, not even after optimization. So flexible area.
How about the A chain? Is it there?
The A chain? It's there.
A chain. Do we see the intramolecular H bond, which certainly is good for binding? since it constrains the flexibility of the residue. So some of you may wonder why we see a duplicate ligand or a seemingly duplicate ligand. This is only because we optimize the ligand a tiny little bit to determine the free energy approximation. So this estimated affinity values. So what enters is subjected to a little minimization. So this is why you see these ligands twice, but this will be resolved in due course when we start to actually work with this. Yeah, what I learned from crystallographers also.
Oh, sorry. There's one comment. I don't know. You should be able to see my screen. If you have problems by not seeing my screen, maybe just re-enter the GoToWebinar interface, and then you'll be able to see it, because I haven't received any other complaints so far. Sorry for interrupting.
Yeah, and I cannot see the full chat as a panelist. I only see the organizer myself chat in this interface today.
Okay.
Yeah. So what I learned from crystallographers is also that we certainly need to be aware that also the experiment is only an experiment. So that means that what we see is an interpretation of electron density. Yeah, there are various ways to actually check the validity or the degree of validity of the structures.
Absolutely. Just kind of like to complete this discussion about this hydroxy group. So what we form here is this intramolecular H bond, which creates a pseudo ring system, which is favorable in regards to the degrees of freedom. So it makes the compound a little bit more rigid and can therefore affect a lot of things. Affinity is one of those, but it's also quite important if you keep in mind if you would like to design something that is blood-brain barrier penetrant, to decrease the hydrophilic area of a compound. Okay, what's next? We have then a pyridine ring, a phenyl ring with a methyl group, so nothing too spectacular here. What I really like is this urea here, which is also at least quite prominent in a lot of kinases for this area, as far as I can recall. Correct me if I'm wrong. And by the end of the day, this is what people actually call the allosteric binding site within kinases. Usually this is also close to the proximity of the DFG motif here. And people are actually debating on whether the selectivity of particular kinases stems from the occupation of this area of allosteric inhibitors, of this occupation of this allosteric binding site of kinase inhibitors, or it really stems from the kinases themselves. So I don't know if people came to a conclusion by now, but at least a lot of selectivity or selective kinase inhibitors contain something in this area. And as we have a trifluoroethyl group in this area, it may be something very lipophilic, but I think we will see in a brief moment. So I would suggest to go with Chin A, or do you have any perspectives?
Yeah, perhaps we can actually use something that I like to use, namely the Hamburg University servers, to assess the validity of the crystal structure.
Okay.
So you may want to make me a presenter, then I can share with you how I usually would get a quick glance on the validity of these structures. So I
should be able to present.
So now it wants me to okay that I actually share my screen. Just a second. It's pretty.
Meanwhile, maybe there is somebody in the chat who also worked with this target class, or at least this particular target. I think everybody had their hands on kinases at some point.
Yeah. So Alex, actually, it would require me to restart the GoTo interface. So perhaps we can simply do that if you are ready to share a browser window. It's actually comparably easy. I should have thought about this after an update. It needs a security setting.
Tell me what to type. You need to select
you need to share your screen again, please.
And there we go.
Okay, cool. So just type into that URL for proteins.proteins.html. plus, please. It's the actual name of the URL, proteins.plus. Hit return. Exactly. So now we are at the Hamburg University website, and we can simply type in the PDB code there to start with, and then click on Go in blue. Hit return for whatever AQG. Yeah, that's correct. So here we go. So now we see pre-selected the 3 ligands in the 3 chains, and we will use one of the ligands, for example, the first, to create a pocket with which we then continue to work. So exactly, click on create pocket for this very first ligand. It tells you about cookies. You can OK that if you wish. And then the thing that we want to use is called EDIA, which is at the second but last. EDIA, structural quality elucidation. So in principle, behind EDIA, there is a voting system. For every atom, we look around, do we have electron density that actually votes for this atom to be validly there where it is? Then comes a little bit of an introduction, plus the literature behind by Agnes Maida, etc., from Ra-Rei's lab. And then we can click on the red EDIA button at the top, at the bottom. Exactly. This will then take us to the actual EDIA calculation page, which says simply calculate. We can click calculate, and now it runs to the service, checks if there is data available, and there is data available. And now if we slowly zoom in, we will be able to see the ligand. What is red is not very validated by the density. What is blue is pretty much validated by the density. So let's see if we see the ligand. I need to zoom in a little bit. There it is. Yeah. Oops. Stand still. Oop. Yeah, because there is a little delay. I just saw 5 ring. Yeah, now it's pretty cluttered on my screen. Yeah, in such a way so that we can follow.
Yeah.
So
like this.
And that's not exactly the coloring that we wanted to, but that's already relatively fine. Yeah. So we see the urea part in the middle. We see lots of blue actually, but we also see red sticking stuff out there. So is that the morpholine in the north? I think that looks like the morpholine ring in the north.
It can be the morpholine ring in the north. Yes, it is.
Yeah. So it seems like that the at a first glance, and you can do this in more quietness, of course. You guys out there can do this in more quietness with these structures or with the structures that you may have at hand. So here we see that partly the extensions are not so safely recorded as we think they are when we just look at a PDB structure. So we can keep this in mind. There is a certain degree of flexibility, maybe at the extensions, at the decorations, if you wish, but the central core seems to be relatively well conserved from what I see at the first glance here. So you might now, if you had more time than we have now, compare this also with the chain B and chain C analogs to see whether something may be much better justified, let's rather say, instead of resolution, because the resolution is slightly below 3 angstroms, I think. But the resolution only tells you half of the story. The other thing is there enough density for the region of interest? So just so that you know, it's an open server, costs you nothing, but careful, if you have proprietary structures, then this will require extra work, for example, porting something like the EDIA server machinery into your organization. Okay, cool. But from my first side, most of the ligand is blue, so that's fine. You see definitely some flexibility around with respect to the chains also that are recorded. But for now, we should keep in mind there is flexibility with the extensions, but overall, here comes the density if you wish to actually see this too. And there you already see that the density gets a little thinner in these regions where there is red, right in front of us, where there is red or pinkish color. Okay, thanks, Alex. Back into CISA, maybe.
Okay, so decision was made. Let's start with chain A. So what happens next is basically the optimization allowed us to detect all proximate amino acid side chains and residues and make them part of the binding site, including the water molecules. What I would like to do is first I will transfer this one to the analyzer mode so I can also see the hydrogen bond network of the compound within the binding site, just to get a better idea. And what I see here is, yeah, this is the most common motif we can observe in kinase inhibitors. We have this particular interaction with the backbone of the hinge binding motif here, basically a gatekeeper residue after this one here. Again, I'm quite surprised about the morpholine, and I would really love to know if the morpholine was actually at the beginning of this whole project or reintroduced at a later point because they needed something. Usually people install also morpholines just to increase the water solubility of compounds. They do sometimes form interactions, but it's not a prerequisite to introduce them. So I'm quite surprised. I'm very surprised. What else do we see?
Alex, what I personally also like to do is to get a feeling for the structure of the entire pocket. So that means but I mean, this is everybody's toothbrush, as Reinhard Albrecht has said back then. But to visualize surfaces, that gives us a much better impression for the start, what this pocket actually looks like, and then switch off the secondary structure visualization, perhaps. So that we get a better 3D impression, not sitting in front of the computer ourselves.
And now calculating the surface. I have to adjust to the new target view control because we changed it from 30.0 to 30.1. So now I want to see the binding site only.
Exactly. Yeah. So
what we can do here is to color it. Color.
Right mouse click on the AQ. Right. Yeah. There. Yeah.
No, it's not working.
Right.
But I have wait. I had the same problem. It is actually a go-to thing. Don't ask me why. It overlays. So transparency settings, and then let's do log p base. Just for the lipophilicity, and there we go.
Yeah, that's what many medcamps actually like.
Okay. What
and
do
perhaps
we see?
secondary structure visualization, we want to switch the chains off because it's spaghetti all over the place now.
Chains. Again, go-to interface problem.
Oh, wow.
No chains. Okay, there we go. Maybe I will also clip it just to get a little bit more of the in-depth feeling. This looks amazing. Okay. What do we see? So this is actually the solvent exposed area, as I mentioned. We have a lot of lipophilic areas and side chain residues in this side, which is quite common for the hinge binding motif, as I can recall. We have the tryptophan here. We have phenylalanine here and here. So it's even more surprising to me that this morpholine actually finds a good spot to bind there. I would expect something, for pi stacking in this area.
Yeah, we could actually try this out in a minute, that we replace the morpholine by something more pi-ish, something more aromatic, so that we create something like a sandwichy orientation between the tryptophan and the phenyl ring. We'll see. Yeah, it is especially surprising since the calculation says that the oxygen isn't even contributing favorably to the affinity, even though this is one of the hinge binding interactions that we would expect to be very favorable.
We also have a cysteine down here, which is quite nice.
Yeah, good point.
So maybe let's do something covalent as well. I would like to do something covalent as well.
Yeah, exactly. So we could use the cysteine as a covalent handle down there. It's true.
What else do we have here? So we have this phenyl ring system with a metal that kind of like really projects nicely into this pocket here as well. Maybe let's turn on unoccupied space to just get a feeling where we still have some room.
Yeah, you can certainly switch off the surface again. It was just meant to help.
I'm actually quite fine with that. I can live with that.
Okay.
It also makes things a little bit colorful, bring a little bit more color into your life.
Yeah.
But everything here is completely cramped, so there is actually not much room available for growing. we will really need to find something else if we really want to exchange stuff here, but let's go with the flow. So this is a very prominent interaction here. We have an ionic salt a salt bridge between the aspartic acid and the lysine here. I think this aspartic acid should be also part of the DGF motif, or is it not? Maybe we'll see, because it's actually the area for this.
An arrangement that tends to be comparably frequent is something cationic in the vicinity of the phenyl ring. Could you measure the distance between the lysine and the phenyl ring of the ligand?
So basically, this is it. I mean,
and the lysine are often floppy, so it could move a little bit.
Yeah, with a little bit movement.
It needs to satisfy the carboxylate, of course.
Absolutely.
That's a strong hydrogen bond, probably. At least it's good geometry, which we can tell by the dark green blobs denotifying the hydrogen bond. 4, 3,
so
3, 8,
what actually is the reasoning behind this one? It's always good to kind of like also communicate why we consider this cation-pi interaction. So basically, if you have this phenyl ring system, you have the pi electrons flying around, and then lysine may stabilize them there as well. This is because it's not like we don't expect you to know this stuff, but maybe some people are just in the beginning of the PhD or students to make it a little bit more insightful for everybody.
Absolutely.
So you have also the urea here, backbone interactions, kind of nicely. Actually, there are a lot of cases where you have something cationic in this binding pocket, so in the allosteric side state may interact with the aspartic acid here, which is also pretty common. Not the case here, but in general, it's possible. And what's really nice here, we have this trifluoromethyl group that actually it's like a claw feet, you know, claw pointing towards the allosteric binding side. Okay.
Absolutely. Okay, so why don't we define as a new sub-challenge then to find something perhaps more aromatic instead of the morpholin? The morpholin is very different than what we usually see in kinases, absolutely admitted, but I, as a drug designer with not so much experience with this compound here, if I put myself into those shoes, I'd be surprised to see something which isn't even contributing very favorably with that interaction that it's supposed to make, but it's binding more or less probably just by the hydrophobic effect, so by displacing water and making it happy around the ligand and or between the two pi systems in the protein.
Okay, I like this challenge. I really like this challenge. So replacing the morpholin. Before we do that, I would like to do two things first. I'm always preaching for redocking. I always preach for doing redocking before you do anything else, because if the docking doesn't work, then you're bound to fail. So therefore, let's maybe do a quick evaluation of the docking. Local machine. And then I don't want to do covalent docking, nope.
Yeah, this is actually something that we haven't done before. Yeah, so it may well be an exercise to see if this now fails, what then? How can we go forward, or if that all comes out good, how we proceed in that case.
So even if you're facing challenges where it doesn't kind of like work out for you, where you don't instantaneously can redock your compound into the binding site, most software, such as ours, also provide you with possibilities to actually decrease the surface a little bit to work with a volume of the heavy atoms of the side chains. So kind of like a little bit more of freedom for the first initial docking of the fragment. So you can kind of like just play around with tolerances there to see if it matches or not. If this fails, I always recommend to do a template-based docking to kind of like place one fragment of the molecule and see if it works, to kind of like just give a guiding light for the docking procedure. Okay.
Yeah. So our docking is an anchor and grow strategy. So we fragment the ligand into pieces and then start with the first piece and build it up from there. Up there we have the input, down there we have the output, and what Alex is currently doing is to color the input so that we can compare what we fed in with what we expect to come out. That is the docking results. So in orange and sticky, that's the pin up there. Sticky now is the original structure, so the PDB recorded crystal structure that has only been slightly been optimized, and now we can compare. Alex?
Okay. So this is the best scoring redocking pose. I would
Aha, and
say
here we
I'm
go.
quite satisfied, luckily.
So in sync with a little bit of leeway from the crystal structure, we see that the morpholin has now been slightly modified, yeah, and now the interaction that we would have expected to be there is there. Alex, sorry, I probably cut your word and spit out the thoughts that you already had. Sorry.
It's good. I don't have to repeat myself. So, but it's like basically now we see actually that the oxygen is not absolutely unhappy there and can actually form good H-bond interactions in the hinge binding section, which is the most important interaction for hinge bind for kinase inhibitors. Good for us. Okay, redocking worked out. We can go through the other poses, but here.
And I should let me stress once more. So this will be absolutely justified by the electron density that we just saw in the other interface. So even though the RMSD is not zero, we have found a pose that makes much more sense from the energetic standpoint and would still be within the density that has been recorded. Derek Lowe once said, "We tend to believe that PDB files are a message from God." And I think this tiny annotation makes a lot of sense, and we very nicely see this now here. Alex?
So the bad scoring poses completely disappear from the binding side. So
yeah,
good. Okay. Second thing I wanted to mention, I always preach that people should actually use what they have on hands to understand the structure. And I'm not a hypocrite, so this is what we're going to do. I would like to actually check for related subsites of related structures that share the same binding site, or at least topologically wise. So now I quickly load the structure into the protein editor and do an CNS search here, just to see if there is anything else available that matches the vibe. So while this downloads, there are two questions. I don't know if we have to address them. Oh, okay. Morphine are actually known hinge binders as for the PI3K inhibitors. I wasn't aware of that. And morphines actually contribute to the selectivity of hinge binders. Okay, I wasn't aware of that, but this is what we are here for. We learn from you, you learn from us. What is to communicate? What's next? So I have this little fella and will straightforward download them to the protein modes to compare them. So this is a compound also for BASF and BUF, sorry. And you actually see there's a lot of common motifs here. We have the same urea, so this is actually already an overlay. We have also the trifluoromethyl group here, and we have a, I would say, more common hinge binding motif. Let's maybe just add this one. I would like to add another two, maybe. Back to the protein editor. Next, I will take this guy here. You will see in a moment why I'm doing this. Again, the same structure, basically, and maybe another one. And this one is, this one is a little bit different. So basically what we have here, pyridine is a hinge binder. Then we have this little fella, maybe to increase the solubility, and an oxam. This is quite surprising, to phrase it mildly. And actually this one does not reach the allosteric binding site down there. Okay, now why I'm doing this, if we now align everything, so 3D alignment of the structures, we can actually take a look at the residues. I have only the binding site, so everything now should be in place. And what we can do here is actually to adjust the RMSD values of the side chains to see if there are any particular differences in the binding site. So what do we have here? Let's look at those things that are different. different
Yeah,
is
so all those
highlighted.
are dimmed, those amino acids are dimmed out now that are further away, or that are within an RMSD of X that we have adjusted, or that Alex has just adjusted with respect to the master sequence, which is the one that shows the little genes pocket icon on the left hand side in that matrix-like display.
Here we see basically, let me just increase it a little bit more. 1 angstrom is not enough. Let's go a little bit more radical, 1.4 angstrom. So here we see that the glutamate is flipping a little bit, but this is pointing outside the binding pocket, so I wouldn't mind.
Mm-hmm.
Then we have a leucine here. Again, same issue. It's not part of the binding site, so I would ignore that as well as a problem.
How about the cysteine? Is that moving?
No, it disappeared now. After 1.4, it's not longer relevant. So it's conserved to some degree. At least the conformation, the watermer. Yeah. So basically, I don't see many problems. I think we can start designing.
Mm-hmm.
Okay. I will just remove them for make it a little bit cleaner for us. Okay. You wanted to have the morpholine removed, right?
Yeah, I would like to search for something novel aromatic to be squeezed into these hydrophobic residues.
Am I allowed to cheat?
Fan man. What do you mean cheat? You are allowed to Caesar.
I'm allowed to
Caesar it.
Caesar. Okay, so what I will do now, I will use FastGo this is our
Oh,
algorithm
Alex,
yes?
that was very fast. So what did you do? All of a sudden, there is a yellow bulb at the top. So you went into the inspirator mode, but since I have it, at least I have a delay, I cannot follow the mouse so swiftly as you perhaps may do it. Okay.
So basically, just for the whole community viewership, if you would like to do something with a ligand in Caesar, you always have to select your ligand and move it to another mode that is dedicated to a particular task. So the inspirator mode in Caesar is what we use to just ideate compounds and look up what can be done, replacing stuff, adding stuff, substituting stuff, all of this. It's now in the inspirator mode, and what I will do, I will define what part of the molecule I would like to keep, basically everything else, and I would like to replace actually the morpholine here. What I will do now, I will actually use special library for this algorithm, which is called FastGo. And this is my new set. So this is the hinge binding FastGo set. Maybe to those people who participate also in the last kinase webinar. So basically, this is a FastGo set for kinase inhibitors for hinge binding motifs. And it contains now 51,000 entries, which is a little bit bigger than it used to be because we also tweaked here a little bit. So it's now way better. What I will do now, I will screen 51,000 of hinge binding motifs to see if we can find something for this binding site. Let's go with maybe 100 results, because there are also some questions, so we can use the time for the calculations to go through them. It shouldn't be long though. So basically, the screening of the fragments is done by now. They are now stored, and now we need to recalculate the binding affinities. Meanwhile, how is this estimated height value calculated? Is there a way to get the docking scores in terms of energy? Markus, this is your question.
Yeah. So height is a development that we did for more than 15 years now in close collaboration with Bayer. Height is spelled Hyde, which alludes to the fact that the mathematical kernel, so the inner kernel, has nothing else than hydrogen bonding and desolvation, Hy and De. Certainly, there are more factors affecting the energy, for example, clash and torsional strain, etc., which is why we have split up the procedure of estimating affinities in 2 parts. The first part is a pre-minimization, where the ligand is subjected to checks. Is there a residual strain with respect to the torsions or any clash, etc.? And only then, when this has been validly resulted, we will show it to the mathematical kernel, which will then determine the estimated affinity using log p increments on the atoms and visualize these atomic increments in terms of these bubbles here. So it's actually published. You may want to search for Heulecke, so R-E-U, or we can type it in the chat. So Alex may want to type it in the chat by Heulecke and Lange, H-Y-D-E. You will instantaneously spot that, if not, just write over to us. And then it has been much refined and brought forward by Nadine Schneider, who is now with Novartis, and again, Gudrun Lange is one of the co-authors, and certainly Matthias Raray, who has been the supervisor of the respective PhD students during this project. So it's not a force field. It's a very different way to calculate the estimated affinity by comparing before binding with after binding, and thus assessing the desolvation penalties or the solvation rewards that you get from placing ligands in certain parts of a protein binding pocket. I hope that this was a coarse explanation of what's actually going on.
Another question
We
is,
can observe if the person who asked is not satisfied, then I can try again.
Another question was, how did I select which chain to pick in the protein editor mode? By the end of the day, it doesn't matter to me in this case, ultimately because this will be just 2 chains from one PDB structure. So just again, co-crystallized structures. It will be more or less the same. I could also load like the second one. For me, it was just based on my gut feeling for this time, because I just want to have an overview of how the binding site may differ in other structures. But okay.
Yeah, that's a great question. What I sometimes do is I try to just search another PDB by not just comparing the binding sites, but perhaps by finding some other inhibitors. So if you go to the proteins mode, perhaps we can actually do this. We still have a few minutes time in the proteins mode. Yeah, exactly. So you need not only type in PDB codes, you can actually also type in B-RAF in complex with, for example, the B-RAF in
B-RAF
in complex with. So typically these are titles, and there you go. So for example, the first one already 3C 4C says B-RAF kinase in complex with PLX 4720. So this will be some other inhibitor that perhaps also gives us inspiration, helps us in finding out where is the pocket probably flexible, etc. Where do we have to pay attention? We don't have to do this now, of course. I just wanted to show you in this challenge setup, this this security-free challenge setup that we've done here, how I'd proceed if I had no clue about what's going on here and I'd get acquainted with the structures. Okay, back to you, Alex.
So back to the results. So again, just to remind us what I did, make it a little bit bigger. We wanted to get rid of the morpholine ring system. And those are now the suggestions from Cosmo. People will be quite happy to see a lot of ring systems here. And we can also, since we have estimated them, we can also rank them again. Some results are not so good, but some are better. So this is the initial compound. Let's mark it with, let's make it hot. Let's make it a fire. Active compound.
I often double click in the name and name it reference, for example, or X-ray or whatever it is, so that you can easily distinguish. Yeah, it's great to have this table here when we cannot distinguish. Perhaps also other properties come into play, maybe solubility and not only affinity. So it seems like the affinity challenge can be solved with the structure that we started with, but perhaps the solubility question cannot, and we can fire up a little bit more of those tables that we have, or table columns in CISA that we have, namely the ones by our friends Optipro.
I would like to color it a little bit more different, just to make it a little bit more poppy. We can also see the differences.
Yeah.
So now this is the first suggestion of an improved compound in this area. Just to kind of like comment on things, we have like a bicyclic system. We have a tyrosine here forming the interaction with the hinge region, which I quite like. What I also like is actually this area here. So we have now this imidazole structure system. It also forms an additional H bond, stabilizing this one. Although we have to comment that in this area, the H bond quality is not in perfect geometry, which we can see with the
lighter
hue of the green. So all in all, interesting. The thing is always when you have kind of like those exotic structures in mind, you always should consider the synthesizability of those, obviously. So maybe we can find also something that's a little bit more accessible and not so exotic. Let's see. Ah, beautiful, isn't it? This is maybe that's so sorry. This one is in pyrazole. I have to correct myself. Pyrazole and imidazole. So this would be kind of like quite easy also to synthesize for my guess. This shouldn't be too difficult as well. Although this is kind of like an aliphatic ring system, which I don't like that much.
Which one? It looks aromatic in 2D.
This
Perhaps
one
it was a little.
Oh, is it?
Flaky.
No, it's at least somewhat aliphatic. So it's sp3 here and sp3 here.
Yeah, okay, it's a little smaller. Okay, good. Cool. Yeah, looks out of plain there. Yeah.
Again, a little bit too exotic maybe, but as you can see here, this quality is better. So also kind of like abusing and kind of like rigidifying this hydroxyl group in this position. Here we get an additional motif, so it's not only like one H-bond interaction with the not only one H-bond interaction with the backbone, but the second one given the amine here. Although one could discuss if this one is metabolically stable. At least if it's terminal, debatable. I don't like this one because it's huge.
Yeah, sticks out of the pocket, as it seems.
There's another problem.
Can we show a few of these columns then by the side? So especially clash, torsions, and I don't know. You may switch on solubility actually. Perhaps we run into areas where we get issues.
Let's see.
So for those who are not acquainted with CISA, we have a statistics-based analysis with respect to the torsional strain that we did back in the years with Roche. That's also been published. And at the bottom of what Alex is showing you currently, we have the octipron properties that help us a lot in having APME always in focus when we're in somewhat later design stages here. Yeah, there you go.
We are now kind of like leaving this sweet spot of rule of 5.
Yeah.
Except for the imidazole. Imidazole is right. Log S, it's decent. I wouldn't complain though.
Yeah.
Though. Okay, so basically from what I see, I really actually like the imidazole compound the most. Where is she? Small one. Maybe let's continue with this one.
Yeah, why not measure the distances now to the pi systems above and below? Have we created something like a sandwich or T-type or herringbone arrangement of the pi systems that looks textbookish?
4.1.
Slightly on the longer side, but still okay.
4, okay. You should also consider this could be like an aromatic H, like that's an identifier for the aromatic system itself. It's not absolutely necessary that it should be kind of like really having those sandwiches there, but just kind of like engage for the initial binding stage. So this is something that MDs can solve, obviously, but I don't think we have the time for MDs right now.
Yeah, also slightly on the long side now.
Yeah, but this is actually
3.7.
not bad. They're not bad.
Yeah, 3.7 up there was. Yeah. Okay.
So maybe continue with this guy. What I will do next is I will remove everything except for the reference and my imidazole. Delete. Okay, next step. What can we do as? So now I would like to switch my databases because my hinge binding dataset is basically containing only hinge binders. So I now would like to work with something more med-chemish, maybe. So I will go
Or
with
not something unexpected. Exactly. Yeah.
let's do this. So this is like our standard set for medicinal chemistry compounds. You will see in a minute that they are a little bit more sophisticated for this purpose. So
How about the
now,
chat? Are people contributing?
are people contributing? No contributions, but the questions were at least addressed. So far we can continue. So maybe just replace this one.
The other way around, right?
Exactly. I would just like to keep the imidazole. Just again.
You are. I got you.
Let's just see what happens.
Yeah. Okay. Yeah. And people could play mix and match later on. Yeah, so while this is calculating again a little bit of background, this is FastGrow. FastGrow has been developed in collaboration with Servier and the Ravai lab in Hamburg. That was a nice project, but a little bit different because it went during the high tides of the pandemic. So Patrick Penner actually was the main contributor here. And it uses bit shifting technologies, which makes it so incredibly fast. Yeah, so we browse these tens of thousands of structures, or even more, within just a few seconds, exploring the conformational freedom while we grow. Alex?
Okay, the results are back in. So obviously we have dropped in an affinity to some degree. Some results are there that are still quite promising. But this is still expected, right? Because
we have got much smaller now, yeah, because we
absolutely.
can just grow into an entire pocket. So we would have to go stepwise, for example, to build this up. You will see that the resulting molecules are now smaller because this is a fragment growing technique that we started from.
So what we get here is again a morpholine, but I don't like the NN bound here.
That's true.
Again, an NN bound. This looks comparatively goodish, I would say. But I would question myself in this case. I'm completely fine with having the fluorophenyl and this phenyl group in this arrangement, where kind of like shifted by or twisted by 90 degrees. I doubt this conformation though. So maybe let's check if for all compounds the torsions. Because from in my mind, they should be at least planar to each other.
Yeah, unless there is a good reason for it.
But let's double check. Okay, where is the bad guy? Yeah, see, it's this one. This always makes sense to double check your compounds because ring systems, at least aromatic ring systems, love to be in plane with each other because the electrons can easily switch ring systems. In this case, this is not even observed. So I wouldn't trust this docking position, or at least its prediction. And again, I always recommend to redock stuff to get to better results. So I don't believe this one. Again, an N-bound. Again, a twisted. So I don't like all of them. So maybe let's help them a little bit. What I will do now, I will actually apply a pharmacophore constraint. Here, place it on this area, make it very, very small, and request any carbon atom in this section. So now I will get rid of the and containing proposals. Do we actually have time?
How many? Yeah, the challenge has already been accomplished, I think, because we found aromatic. We found lots of aromatic that would do the pi arrangement in some way. But how many are we now browsing with FastGrow?
This is the MedChem set. So per default, without any addition, Caesar comes with 12,000. This is the larger set that can be downloaded from our website, free of charge, obviously. What's behind those 120,000 fragments are additional 10 confirmations of each of the fragments. So basically, we are now browsing through 1.2 million confirmations within what was required for this, like 1 minute with rescoring. Okay, what do we get now? Okay, see, so this is where I become more happy. We also have now a green torsion here, so things start to light up. Red torsion though, red. But we actually can introduce a link system. So this is what the phenylalanine, the pyridine group was in our starting point. I really like this one. How do you feel about this?
Yeah, let's check the solubility again. We have one charged group or chargeable group, at least.
We need to recalculate everything. Okay.
This, by the way, can be automated, but it seems like Alex and both we like to do things stepwise to be more didactic when we talk to users.
And I don't. Here we are. This is this one. Solubility, wow, debatable. Debatable. We can reintroduce solubility. But actually, no, I really like how it really kind of like just complements the binding site here. As you may recall in the original compound, we can also just, again, make it sticky here and color it in orange, maybe. Not in orange.
Yeah.
Let's do ice blue.
Yeah, so you see how we follow the original shape a little bit, but also I think this this challenge serves at making clear that a quick shot at something will not be done within an hour. Yeah, so I think we conveyed that this is kind of a very discussion-rich approach that profits from multiple people taking part, and that we can juggle ideas, we can produce ideas, etc., relatively quickly. But the sub-challenge of getting, or the sub-challenges, I should say, of getting acquainted with the complex, getting a feel, estimating its trustability, and generating something as a replacement for the morpholine was easily achievable, I think. Yeah. So now at 16:59. Yeah,
maybe
put this back in your hands now.
Before we end those things, I would actually just show from a perspective, you can also just keep on going like exactly like this. So again, you can, if you really like this structure, you can just say, well, I would like to continue growing. This one, and say I would like to keep going from this nitrogen here. We don't need this pharmacophore constraint any longer. Then use the same library for another 100. The variation of those kind of changes kind of pretty nice and really like it. We will find something that's the best thing that happened to me right now is we can find readers that is a T4 room fit to the allosteric binding site. And after that, I would like to do a quick and dirty covalent test. Really quick and dirty, just to get ideas. So, okay. This would be the new proposal. Again, a little bit of nitrogen, but with a pharmacophore constraint, this is something we could actually solve maybe. And in this case, we are also somewhat able to project into the allosteric binding site there. But see, it's not exactly where it's supposed to be, but at least for the second structure we've loaded in, not the original one we're working with right now, but one from the Sienna search, we're actually also finding the T4 image group. So I'm at least somewhat satisfied.
Again, an N here that we get rid of. Yeah.
So about docking. So let's clean docking up. I think I have a set prepared somewhere. Give me a second. This is something of my side hustle. Very quick. So basically, this is a very quick and dirty approach that I used for some internal testings. These are just covalent warheads containing an input 2 group, a phenurine system, and some pyridine derivatives. And there are a lot of them. So this is kind of like a very small set to just kind of like sample potential warheads for a binding site. What I would like to do now, no, that's wrong. Binding site. I need to focus on binding site. And there was a cysteine somewhere, right?
Yeah, southeast. What was it?
There she is.
531, I think. Yeah. Can you label it? Is it that one? But I think it's the only one.
The question is,
531. Yeah.
why can't I select this one for covalent docking? Where are my?
Could be.
Where
Is
are
it
my?
not? Is it not part of the binding site for whatever reason?
There she is. Okay, reloading did the trick. So now I've selected cysteine here. You're just renting the mode, and then covalent docking. So it's like just a sample if there's any warhead.
Yeah, that's all that.
Potentially. Didn't take too long though. Almost there. Good things, the fragments are quite small, so it's can be done in a manageable time frame with 165 informations each.
Yeah, not using the cluster, so it's just running on your laptop, I think, or on your machine.
So if you have something on your hands for a fit PDB, you can all reach out to us. Maybe we will invite you next time for the next session. And then you can comment on things and provide us with additional insights. Almost there, almost there, almost there, almost there. So basically, this is also quite novel to CESA. It was introduced in 13.1. The warheads that are now present in compounds are automatically detected and transformed into their bound form. So no workarounds required. And those are the results.
Yeah, and the chemistry is also automatically performed. So the warhead is dewarred and connected to the cysteine or the other selected amino acids. The respective chemistry is automatically performed.
So what I really like in some cases that we really get those additional hinge binding motif interactions with the backbone on top. I wouldn't trust hydroxyl groups here because in most cases they are usually only donors. I don't know how acceptors would behave in this case. But as you may see, there are some proposals how to actually maybe introduce a covalent warhead there.
Yeah, so now we are in idea stipulation mode a little bit more. So with which you can find out.
I really love this one. I really love the sulfoxide acrylamide here. What is also possible to do, you can also apply pharmacophore constraints for the unbound compounds, for the untransformed, to see what could actually match there. You can also quickly do this as a last feature. What I mean by this is you can actually apply a pharmacophore constraint in proximity of the cysteine. Let's do like a bit more generous, maybe 3.5 angstrom. And we would like to have the covalent warhead placed in the proximity there. And then let's do the normal docking, the standard docking. The idea behind this procedure is actually to see can a covalent warhead also be can it also bind in proximity to the cysteine without being bound? It's like is it the egg or is it the hen? What was first? Is it really the binding of the ligand itself and then the covalent bond, or is it the covalent bond and then the ligand binds and binding pocket? Okay. Meanwhile, Markus, while we are in the last few minutes of this session, question.
We learned a lot again from looking at the ligand or protein ligand at the complex structure before you actually do anything with it. So proper analysis and we actually operate. These are very important steps. And I like your idea of checking for staining sites using the Hamburg servers. We learned that we should not leave out of sight the properties beyond affinity. So that means torsions, clashes. You highlighted the NN, the unwanted NN connections here and there for synthetic aspects. So it became very clear, even within these 70 or so minutes, that drug design is a multi-parameter optimization, and visualization helps a great lot. It makes a lot of sense to actually put yourself into the room with a few of your peers and discuss, as we just did here, ping-ponging. That definitely helps a great lot. I think it was a good exercise, and I'd be very keen on having more reactions in the chat or in other forms of contributions. We'd love to hear from you guys.
Absolutely. If it was too boring, also mention to us so we can adjust accordingly. But if the reception is great, then maybe we have a second or third or even a whole series of those kind of things. I would also like to work with EPCR if I'm honest, if I'm allowed to be honest.
Yeah, you guys take the next challenge or challenge, Alex, with your PDB.
For the last few calculations.
Yeah, it's all done indirectly here.
It is also the purpose of the whole thing.
It will be a matter of just a few more seconds. Exactly. Yeah. To really give you an on-the-fly, real-life impression of what this is like. So I've been visiting various companies that actually use CSR, and I've always been impressed by a setup where, for example, 12 people actually, the department or the project team, or a dedicated project set together, and then somebody says, try out a CF3 group back there, or did you ever try to connect to a morpholin, etc. And all these things can then be tried out interactively and can be discussed in these circles. Makes a lot of sense.
So there we go.
Also with
yeah. And this is basically what I was hoping to see. You can also place some sort of covalent warheads, in this case, something that also interacting or forming a lot of interactions, hydrogen bond interactions in the binding site here. And this is a very valid covalent warhead. And there's even I think there may be even room for, won't be room for a fluorine though. Yeah, but this is perfect for ideation, just kind of like gather new ideas how to actually maybe introduce something new to a compound and make it more interesting. Okay.
Yeah. Alex, did we leave any questions unanswered so far? Because I don't see the chat. I don't want to leave anything open.
So basically, there's one question. There are a few requests about our emails. But since we have your emails, we will simply reach out to you afterwards, so we can discuss things. Again, your last
generic. The generic email would be contact@BioSolveIT, of course, in case you have general questions or would like to reach any of us. That will be forwarded. Alex?
Absolutely. Last but not least, I would just maybe give you some more minutes if you would like to say something, ask something. Meanwhile, I will use this opportunity. It's really good too. It's surprising. We'll use this opportunity to announce the next webinar. Will be about the Infinity 6.1 release, which will be up in 2 weeks max, I guess. And also we cap off the 13.1 release, so I will keep you updated about the next date. And then let's see what new questions may be there. Seems like there are no questions we have to address live so far. So therefore, I hope you enjoyed it. I did, to be honest. I don't know about you, Marcus.
I hope I wasn't too strict.
Never. So with this, thanks everybody for participating. It was a pleasure. Reach out to us if you need anything and speak to you soonish. Thank you, everybody.
Absolutely. Take care.
Good
Thank
time.
you. Bye-bye. Cheers.